Okay, we'll start with just a quick review of the respiratory epithelium. This is actually a real picture from the Mount Hood Histology Library, so you can see that it's pseudostratified, pretty lengthy cilia at the apical surface, and just remember, it looks stratified, but the nuclei are at multiple levels, so that actually designates it as pseudostratified. For the trachea, the keys to remember are the complete intact ring of hyaline cartilage and the posterior trachealis muscle. Keep in mind this structure is very large and we won't be able to see the entire intact trachea under the microscope, even under the lowest magnification. Here's actually a real example of a trachea. They've missed a little bit of the hyaline cartilage ring, which is just an artifact of the cut, but you can see the posterior trachealis muscle on the left. You can see the purple C-ring of hyaline cartilage almost extending all the way around the rest of the structure. For a bronchus, and for our purposes we will not make a histological designation between primary, secondary, or tertiary, but the idea is that the C-ring of cartilage has broken up into individual plates. Keep in mind these plates can look more on the pink side, as in this specimen, or more purple and dark, as in the last specimen. So these are all just bronchus. For a bronchiole, there is no hyaline cartilage. In fact, it's been replaced entirely by smooth muscle. Occasionally you will see that the cuboidal epithelium of the bronchiole is disrupted with an alveolus, and because that alveolus is capable of gas exchange, we designate that particular area a respiratory bronchiole. Here is a really good example of a terminal bronchiole indicated by the black arrow, basically branching into the alveolar ducts and sacs. So you can see there's quite a difference between the thickness of the epithelium of the bronchiole, which should be cuboidal, and the alveolar ducts and sacs. On the right is a bronchus, and there's a single plate of hyaline cartilage on the upper right, and you can see, even though it's a little bit blurry, that definitely is a hyaline cartilage plate, so that would make that a bronchus. Here's another bronchiole. You can see really nicely the dark contrast of the cuboidal epithelium with the surrounding simple squamous. So it's a really nice slice because it goes right into an alveolar duct. You can see sacs there at the very end of that structure. Just real quick, an alveolar sac is like a cluster of alveoli. So if there's more than one, we just designate that as a sac. If you take a real close-up look here at the respiratory membrane, you see that there's several types of cells. Type 1 are just the simple squamous cells themselves modified for gas exchange. Type 2 are the surfactant-producing cells, which are also integrated into the basement membrane. The other type of cells that you may or may not see are sort of independent from the respiratory membrane. They're kind of free on the surface. Those are the cellular macrophages, or sometimes called dust cells. Here's another example. You can see the blue arrow indicating the type 1, which is just simple squamous, and then the rounder type 2 are indicated by the green arrows.